In this video, we're going to take a look at bacteriophages, sometimes just called, or usually just called phages, which are the viruses that attack and kill bacteria. So bacterial replication occurs in 5 steps. First of the steps is adsorption. This is just a chemical attraction that causes the bacteriophage to adhere to the bacterial cell wall. Second is penetration, where the virus or the virus's genome is injected into the bacterial cell. Third, synthesis, there we are, which is when the virus takes over the host cell's all of its metabolic activity and forces the host cell to synthesize the viral proteins that form the capsid around the DNA and the nucleic acids that are found inside of the viral particle. Third is maturation. This is the process of assembling the virion particles, the virus particles themselves, the virion, from all of the newly synthesized nucleic acids and proteins. And then finally, release, which is when the host cell dies, and usually that's what we see, is that the host cell dies, bursting open, and all the viruses are released from the bacteria. Sometimes, I'm not sure if this ever happens with bacteria. It can happen that in animal viruses that the virus particles will be shed slowly from a living cell that remains living for quite a while. But I think with most bacteriophages, the virus particles are released as the cell lyses and dies. The bacterial, so this process can be used by us to treat bacterial diseases, because generally bacteriophage infection in bacteria ends with the cell lysed. Here is an example of a lysed cell. So we have these rods that, as the bacteria are filling up the cell, the bacterial cell just gives up the ghost and explodes, leaving or releasing all of the phages. And so we can use phage therapy because this rapid production of phages, it can take between 20 and 40 minutes to go through this cycle of invasion, and then the lysis, invasion of the cell by the bacteriophage followed by lysis. So we can very, very quickly outpace bacterial production with all of the phages that are being produced by the dying bacterial cells. And then as the target bacteria become depleted in the body, then the remaining phages, they won't have anything to reproduce on, and the immune system just removes them from the body. So it's kind of a cool setup that you have this killer particle that will go in, destroy all the bacteria, and then the body just cleans up all the stuff, and we don't have to worry about harming other cells around the bacterial infection. Viruses are very specific to the pathogenic bacteria that they're used to be treated. They are specific to those bacteria, not to any other body cells. And so this shows huge promise in the ability to fight infections, including, and this is really exciting, including the ones that are caused by antibiotic-resistant strains and the ones that are caused by biofilms. Resistance does get, eventually the bacteria develop resistance to the phages, or can develop resistance, but bacteriophages are fairly easy to develop. And so this, as we think about entering the era of designer medicines, where we fit the medicine directly to the person who's taking the medicine, excuse me, and to the specific disease the person has, phage treatment, phage therapy has great promise. The Soviets are way ahead of us. Or I say Soviets. They were developed heavily. You know, the Russians put a lot, or the Soviets put a lot of emphasis on developing phage therapy. So Russia has the leg up on us. We did not take that same kind of really aggressive approach toward phages, and so in using them as therapy. So there's a lot to be learned, especially if we can figure out ways to share these technologies with other countries and other professionals. Okay, so let's take a look at T-even phages, T2, 4, and 6 phages, which is just a classification of phages. We're not going to worry too much about what that means, but we're going to use them as our examples for how bacteriophages do their work. So adsorption is the first step. Absorption is a chemical attraction between protein recognition factors that are found on the tail fibers. The tail fibers are these little dudes right there, where the bacteria, they look like little lunar landers to me, where the tail fibers are attracted to receptor sites that are found on the bacteria on its cell wall surface. And so we see this chemical attraction that attaches the phage, the capsid, the external structures of the phage, onto the cell's surface. And as those tail fibers attach, they bend, and so this portion here is referred to as at the bottom of that portion, of that little stem-looking portion, is a structure called the pin. And so as as those tail fibers flex, they bring the pin into contact with the cell surface. So T4 phages, they adsorb onto the cell wall of their bacterial hosts. Other phages are capable of absorbing to flagella or to pili. So there's a lot of different places that different bacteriophages will adsorb to. So that's adsorption. Next comes penetration. So these bacteriophages carry with them lysozyme. It's a protein that can then be released from the tail of the phage, and that lysozyme will weaken the peptidoglycan cell wall enough so that when the tail sheath contracts, that will force the hollow core of the tube of this sheath. It'll force that core to come into contact with the phage wall, cell wall, and then through the cell wall to the bacterial cell membrane. And then the virus can push its DNA through from the phage head, this portion up here where I'm throwing things around. This portion here where the DNA is contained pushes it through the tube of the tail and into either the periplasmic space that is between the cell wall and the cell membrane, or all the way into the bacterial cell. So it may only enter the periplasmic space, but recall periplasmic space is very metabolically active too. So we can do a lot of work there in the periplasmic space. The capsid of the phage, this head and tail itself, will remain outside of the cell. It's only the DNA that gets injected either into the periplasmic space or into the cell itself. So the next step then is synthesis. Once it's inside the cell, then the bacterial DNA is disrupted, so it stops being used to make proteins, and instead the phage DNA is used to make, if you recall from our discussion, messenger RNA, the RNA that then goes to the ribosome to be turned into proteins, to be translated into proteins. So we translate that mRNA into proteins to produce the capsid proteins and any viral enzymes that are required. And there is also within the virus DNA polymerase, it carries the instructions to build DNA polymerase. And DNA polymerase is an enzyme that will assemble new DNA lengths based on the DNA that's found in the virus itself. So during synthesis, we make all the components required to build a virus particle. Maturation, then, is the process of assembling virus particles from all of these newly built materials. So the capsid proteins assemble into this polyhedral head, this kind of oddly shaped head there. That's a polyhedron, is the shape of that head. And then the viral DNA is packed inside of it. The phage tails are also assembled from the base plates that are built, the sheaths, and the collars. And once that phage tail is assembled, it's attached to the head. And then finally, at the very end, the tail fibers are added, and you have a mature, and it is now pathogenic bacteriophage. Then last of all is the release of the phage from the virus cell. So as the cell fills up with these matured phages, then lysozyme, again the enzyme that the bacteriophage DNA has instructions for making. So the cell produces a lot of lysozyme. That will then break down the cell wall, which, as the cell lyses, will then release all these phage particles. So a T4 cell, or these T4 phages, I shouldn't have called that cells. Sorry about that. It's a T4 phage. Those T4 phages are referred to as virulent phases or lytic phages because they always wind up with the cell lysing. So if something is virulent, that means it's very dangerous, it's poisonous, it kills. And lytic means that's a reference to the process of lysing, the cell breaking open. And this cycle that we see here, this round and round and round of infection or adsorption, penetration, synthesis, maturation, release, that cycle is referred to as the lytic cycle. The burst time, the time from adsorption to release, is usually somewhere between 20 and 40 minutes in these T4 cells. And we can talk about the number of virus particles, the number of virions that are produced during one lytic cycle as the viral yield or the burst size. So you can hear either of those two terms. So viral yield or burst size is just that's the number of virions that are released at the end of a lytic cycle. So somewhere between 50 to 200 new phages are released with each lytic cycle. Okay, so just like we saw with bacteria, there's a phage growth curve as well, and it's described as the replication curve. And it just it describes how many virus particles are found in a volume of body fluid. And there are two phases. I can't get those two words separate this morning. There are two phases to the lytic cycle. There's the eclipse phase. This is the period after penetration, but before new virus particles are released. So it's a time during which viruses cannot be detected because they are found inside of the cell. So this is a 20 to 40 minute period. That's the eclipse period. The latent period, that's the time from penetration, so again, the beginning of the eclipse period, all the way through the time that the phages are released. So I described that badly. Let me go back again and make sure that I'm getting this right. Okay, there we are. Eclipse period is penetration from the host cell through to the time that synthesis of new virions is occurring. The latent period is the time from penetration to the release of new phage particles. So during, at the end of the eclipse period, even though virus particles haven't been released, they are built and they are hanging out inside the cell and they're accumulating inside the cell. So if we go and break up the cells, we could find virus particles. During that eclipse period, we don't have any fully assembled viruses made. So we see penetration, but no production of assembled viruses yet. The latent period includes the eclipse period plus that time after the time of maturation and release. Okay, hopefully that makes sense. Okay, so how can we determine phage concentration? It is usually done with a process called a plaque assay. So you start with a suspension of phages that you don't have a known concentration of, and just like we saw with bacterial counting, you would do a set of serial dilutions, and then you would spread the most likely appropriate countable dilutions onto it. You would spread it onto, along with broth of a susceptible bacteria, onto an agar plate, and then you would select the plates with a countable number of plaques, count those plaques, and multiply by the dilution factor in the dilution series. If you have done a good dilution, then each plaque would represent just one virion, one virus particle, and its replication, killing off the bacteria in that area of dead zone. So it's the dead zone that you count as a plaque, and that dead zone should correspond, if you've done your dilution series well, should correspond to just one virus particle. And then you report the information that you have about it as the number of plaque-forming units. Okay, so not every phage kills or causes a lytic cycle and destroys the host. There are some phages that, rather than immediately causing the destruction of the host, actually just inserts its virus DNA into the bacterial genome and allows the cell to continue living and reproducing, but each offspring cell contains this prophage, this set of virus protein-making instructions. We refer to these phages that do not initially cause a lytic cycle as a temperate phage. So they exhibit what is referred to as lysogeny, which is the stable relationship between the bacterium and the virus, where the bacterial cell is allowed to go ahead and continue its growth process. And so the lysogenic cycle then is this portion of our life cycle here, where the bacteria with its prophage in its genome will continue to grow and divide, grow and divide, grow and divide, grow and divide. Interestingly, when they are in this lysogenic cycle, the bacteria will become resistant to reinfection of any of the same group of phages. So only one bacteriophage is allowed to place a prophage into the genome of this lysogen, this bacteria host, plus its temperate phage. And so it prevents the adsorption of any other of the same species of phage. So it can last for a long time, doing this stable, as a lysogen, doing this stable lysogenic cycle, and remaining, and experiencing lysogenic conversion. Now, lysogenic conversion does not protect it from other groups of phages. So if a different species of phage tries to absorb to it, that phage will be able to absorb and also infect it. But any phages of the same species of that virus will be unable to infect the bacterium. So this lysogenic cycle continues, producing more and more offspring cells through binary fission, excuse me, through binary fission with the prophage in the DNA. But at some point, induction will occur. Induction is either a spontaneous event where the lytic cycle begins, or it can be some sort of outside stimulatory thing that can happen. And so, it may be because nutrients are becoming decreased in the environment that the bacteria is living in, or there may be chemicals that are present in the environment that are going to kill the bacterium, but the prophage recognizes those conditions, and as conditions become deadly for the bacterium in some way, then the induction would occur, and the bacterium moves out of the lysogenic cycle here and moves then into the lytic cycle, where it begins to synthesize the viral DNA and viral proteins. There's maturation of the bacteriophages inside the cell, and then the cell bursts, releasing the bacteriophages. So the majority of bacteriophages will undergo lysogeny. They're capable of doing that, but will eventually enter the lytic cycle over time.